Okay, I don't think you need to know anything else about me. It was in the bio. What's more important is my message rather than me. And thank you for indulging me to share with you a little bit, another sermon that I'll do on fungi. This time it's on spore dispersal because I think it represents some really interesting stuff where physics, chemistry, biology and evolution and ecology all come together. So I'm hoping I'll get some of you motivated and excited by this after the talk. And not throwing fruit at me. Okay, so how fungi travel in time and space. It's sorry, I've got to remember I can't move it on with my clicker. I could get Wayne to do it. Okay, so fungal spores, in terms of the fungus, thinking about the fungus, they represent the termination of the developmental reproductive stage in the fungal life cycle and the start of a new generation. They're a reproductive unit capable of being dispersed over great distances and waiting out adverse conditions. So that's the role for the fungus. But the role for us, I suppose, for mycological society and people going on forays, is what are you doing when you're collecting them? You want to identify, you're actually intimately studying the spores and the fruiting bodies in which they are. And so from that perspective, from the human perspective, that's really important. The fungus actually doesn't care whether you know what it is, what its species is or not. So it doesn't care that its spores are meant to delineate some ID. Really, its role is, as we've said, it's a reproductive unit. So bear that in mind, that sometimes you get to see that you'll hopefully at the end of this talk, you'll get to see why there's a fair bit of confusion on the morphology of spores and why you actually seem to have a lot of convergent evolution and why spores and fruiting bodies are not actually that easy sometimes to delineate species, which we've really found now in the molecular age. We find that things we thought were closely related are actually quite distant. Okay, so it's interesting, this statement from Kendrick in his textbook. So the key reason for the success of the fungi is their ability to produce and disperse vast numbers of tiny but often highly specialized spores. By sheer fecundity, fungi make sure that whenever and wherever a new food substrate becomes available, they're there to exploit it. So if you wonder why they're so successful, they're there. Okay, so what do? They produce spores in great excess, because the laws of probability are working against the success of any one single spore. So the majority of the spores are wasted. It's not a problem. If you think about the giant puffball, Calvacia gigantea, it produces 7 times 10 to the 12th spores, basidiospores. I don't even know what you'd call that number in a basidio cup. And Buller back in 1924 had the time to calculate that if each spore were to germinate and produce an equally large basidiospore, the combined mass would be 800 times the size of our planet. So thank God very few spores actually make it to reproduce. Okay, so just to take you back on the fungal life cycle to remind people, some may or may not be familiar with it, that most fungi they've got two parts of their life cycle. They have an asexual cycle and they have a sexual cycle. Some actually only are stuck in the sexual cycle. They've never been found to go into the asexual. Some are stuck in the asexual cycle, and some move between them. In the asexual cycle, so the mycelium, which is the body of the fungus, or the soma, it grows through a substrate, so you don't see it mostly. It will produce asexual fruiting bodies when the conditions are right and it wants to exploit that environment really quickly. So the asexual fruiting bodies are really, basically the spores, they're very, very simple. Not a lot of work has gone into a structure. Their immediate colonization, if you think about that's what their primary role. They produce conidia. I'll talk a little bit about that more. The sexual cycle, so you have a mycelium, the threads, which are growing through a substrate. If they grow in the same substrate as a mating type, as the opposite mating type, then there will be a sexual union. They'll then have 2 sets of chromosomes in them instead of 1. And then they will then produce a fruiting body. And those fruiting bodies can be quite complex, given that it's simply a thread-like organism that can produce these 3D structures. So the fruiting body can be very complicated when you start to think about it, and they're actually quite interesting biomechanical devices for the dispersal or the discharge and dispersal of their sexual spores, which then can germinate and go back into an asexual cycle or a sexual cycle again. Okay, so just recapping. The types of spores and roles. So we have asexual cycle, and for the higher fungi, which are ascomycetes and basidiomycetes, we call them conidia, because the conidium singular is a spore that's born on aerial hyphae. Now remember, I'm saying aerial hyphae, so that has to get it up off the substrate. And for the lower fungi, which I tend to not do, I'm making an exception tonight, I tend to ignore them. Their asexual spores are called sporangia spores because they are produced in a little structure called a sporangium. Okay. The sexual spores, and again, I'm really being very simplistic here in my taxonomy, because the ascospores and basidiospores are the sexual spores of your higher fungi, the ones which produce cross walls and they can produce 3D structures. Zygospores are from the lower fungi, but there are a whole lot of other groups of lower fungi, depending on which classification system you've read lately. But most of them, they're not worthy of talking about in this talk, so I'll just skip it. Okay, it's a cop-out and a half, wasn't it? Okay, the study of spores is also the study of evolution and how they've evolved to deal with different environments and different situations. So the fungal diversity, what's that thing doing there, Wayne? Can we get rid of that message? Thank you. Okay, fungal diversity is revealed through the size and shape of the spores, their development and mechanisms of dispersal. And that dispersal could be passive by airflow or raindrops or via these biomechanical devices to launch spores into the air, where there's been quite a lot of really interesting evolution that I hope to talk about, help you to understand tonight. And these mechanisms can produce some of the fastest movements in nature, believe it or not. We may be only talking a very small scale, but they can be very fast. And because of that, annually, fungi release millions of tons of spores into the atmosphere, something I'll come and talk about right at the end. Okay, so before dispersal, because it said the spore is there for dispersing, we must have discharge from certain types of fruiting body. And it's very interesting, if you think about something that's growing in a substrate, and it's got threads growing through the substrate, what sits above most substrates and most things is a boundary layer, okay? And that's a layer of still air. Now, we don't normally think about boundary layers because they're small, but if you think about the spores, which I forgot to tell you the size, that the most fungal spores are about between 1 micron and 20 microns on average. You do get some which are producing big ones up to 50, 100 microns. They're more the exception. Most spores around about 1 to 20. And so boundary layers are an issue. So if they're being produced right on the hyphae, or the threads, just about even on the surface substrate, they've got a boundary layer which is going to be laying, pushing down on them to stop them moving. So what have fungi had to evolve? The first thing, a lot of their structure is actually this problem of evolved to shoot spores through that boundary layer. If they're not doing that, then maybe they've co-opted another agent, such as water, wind, or animals, to help them get through that boundary layer. Because essentially they need to get up into the turbulent layers of air so they can get dispersed. Okay, so just looking at the diversity of mechanisms of spore release through the boundary layer, it's still there. So the grey shading here will represent the boundary layer you may see in different fungi. So you have, this is a typical, some asexual and nectaria, where you get water splash, will splash the spores off the surface. These are, again, I think they're the lower, actually, I think that's an oomycete then, so it shouldn't even be here, but we do still treat them as fungi. So you can see that their structures have grown them up beyond that boundary layer, in this case that we picked up. And some of these, they actually have cells that round off to push them up. And then we have a whole series of, your mushrooms will usually grow, the stipe takes the cap right up above that boundary layer. You have puffballs grow above the boundary layer, but ascomycetes often are underneath that boundary layer, but they've evolved shooting mechanisms. So that's some of the things I'll start taking you through, these actual devices that we see in different fruiting bodies. Okay, and so you can see clearly in this diagram here, you've got a bracket type fungus here over on the tree, and then you have a typical mushroom, and here you have a typical ascomycetes cup. You have the ground, but you can see that down close to the ground, you've got that lamellar layer with the boundary layer and the bottom lamellar layer where you're having very little air movement, particularly thinking in terms of forests where there's little air, and your turbulent layers up high, that's where you want your spores. So A lot of the structure in these is going to be getting some up into that structure, up into that layer. Okay. So spore discharge can be either passive, in which case the fungus is co-opting just nature, air currents or raindrops, or it can be active. And the active ones are where we get some really interesting physics or an interesting biomechanical devices. So you can have explosive action, or you can have eversion, and you can have tensile water with giving catapults. So I'm going to take you through a few examples of that. And I'll note that this is not a totally comprehensive. This is really just like an introduction with some of the more interesting main examples to take you through to get you interested. Okay, so this, once you've got discharge, you then have the spore dispersal, so the journey can begin. So air currents, so once the spores are caught by wind, they can be carried very long distances. And they can actually, if they believe they can stay up for days, weeks, months. Some people even speculated years, but I'm not too sure they'd be viable by the time they came down from years, because you've actually got the problem of UV, and a lot of spores aren't going to have enough resistance to UV to be viable for that long. But certainly, if you think about islands that are formed from volcanoes that become colonized, a lot of the fungi are going to get there through air currents. They go to, and I think often because I've been over to Norfolk Island, which has an incredible microflora, and most of that would have had to come in by the air. So I find that a lot of the stuff you see there is similar to here, but different, which is what you'd expect. They're coming in the air over millions of years, and then they're having their own little bit of evolution to their own ecology there. And of course, water and animals, and the animals, whether it's purposeful or they're just inadvertently carrying the spores on the surface, are very, very important agents for dispersal of the spores. Okay, so let's start with the passive discharge, so air currents. Air currents are the main way that dry spores are spread, and that is most of your asexual spores. Most of them are dry, and they're going to produce spores which are just sitting up above the mycelium on a conidium. So it's got a conidiophore that lifts it up. And their spores are interesting because their spores usually are coated with a substance called hydrophobins. And as you say, you know, hydro meaning water, phobin meaning it's anti-water. So it's not water loving. And those hydrophobins keep those spores dry. They actually help the spores separate. And the fungi I work with are the entomopathogenic ones, particularly Metarhizium, are extremely hydrophobic. And so they've got a lot of hydrophobin rodlets on them, but it's also interesting that not only does that help them get puffed up, because it's really, when you're working with a lot of Metarhizium spores, it doesn't take much in a lab to, you know, breathe over the colony. You've got spores going everywhere, which can contaminate plates very easily if you're not careful. But those hydrophobins also help the spore deposition, because those fungi I work with evolved to work to invade insects, and insects usually have a layer of waxing, a cuticle. So hydrophobins are perfect to make them stick to that as well. And then you get hydrogen forces, and then you get germination following after that. Okay, so a lot of asexual spores, air currents. So some gastromycetes, say your puffballs, they can use air currents for the discharge, but it's not as much commonly as what I'm going to talk about next. And some cups, like this is an acervulus, an asexual thing. And I think some of your little cups in your ascomycetes, the air currents, because cups create turbulence inside them. A flat surface doesn't, but the cups can. So air currents can be used for that passive discharge. Air currents are far more important for dispersal though. Okay, now the passive discharge raindrops, these are really important. So the gastromycete group, which are puffballs, earth stars, birds' nests, raindrops are extremely important because they don't have the same mechanism that most other basidiomycetes have. So when you see something like this puffball here, where you have a thick peridium outside of the spore mass in the gleber, raindrops landing on that surface act like little bellows to push the thing down and back and down and back, and those dry spores will puff out. Earth stars work similarly because they've got that little puffball structure in the center, but their star is really a covering that bends back on itself to push it up where it's going to be more effective. So if that little puffball section is up higher, it's even more effective when raindrops hit it to puff out the spores. And asexual and sexual spores produced in slime, again, a lot of the insect pathogens will produce spores in a slimy head. Raindrops are important, and some ascomycetes, raindrops do play a role there. Okay, so there are our passive things. Okay, so raindrop dispersal and bird's nest fungi. Now, I think how many people have never seen a bird's nest fungus? Okay, next time it's wet and you see a whole lot of mulch, look carefully. These little guys are about 5 millimeters across, and they often come in masses, and they're really pretty when you see them. They literally look like little bird's nests. So you look up closely, because imagine these are quite small, and you've got to look carefully, and they blend into the mulch of their colour. When you look closely, they look like they've got little eggs in them in each of these cups. And those little eggs are actually what we call peridioles. They're a structure that contains all the spores. But underneath those peridioles, you've actually got this long thread, which is curled up to underneath it, called a funiculus. And at the bottom of the funiculus is a little sticky end called a hapteron. And so what happens, I'll take you to the next slide, where a raindrop comes in, it's hitting that cup, and the raindrop is usually not, big raindrops aren't much bigger, sort of smaller than the actual cup. The cup, interestingly, has a design that the walls of it are about an angle 60 to 70 degrees to the horizontal, which has been shown experimentally to be perfect for breaking up that droplet, pushing it down, and optimally getting under and pushing those peridioles up. So the peridioles go up, and as it goes up, the funiculus unwinds, and the hapterons at the end, so go down to this bottom picture here, that as it flies through the air, the sticky hapteron will actually impact on a leaf blade or something. When it does that, it pulls the peridium back, which winds around, and now you're stuck on a grass ready for some animal to come along and eat, and the life cycle then goes into the animal, dispersed, comes out in the manure, and it's got a perfect place for it to continue its life cycle. Okay, so that's one. Now, let's get into the active discharge. So the active discharge, and of course, this is the group that most people who go on forays are going to be encountering with your mushrooms. Okay, so the active discharge, the blister spore discharge, is actually a surface tension catapult. So it's using surface tension to catapult those spores. It occurs in all basidiomycete groups, except the gastromycetes, which I've mentioned just before. And it's best illustrated in the mushrooms. So we think about the mushroom shape, and everything about the mushroom has evolved, is about this blister spore to be successful. So we have a cap, it's a little umbrella with gills underneath, a stipe that can be right up off the ground or short. It can be, and all of this comes into it. The gill is actually a whole lot of tissue which is folded to give you a maximum surface area. On that surface, each gill, you have the reproductive structure, so you have a basidium, and then on each basidium, you have 4 little points called sterigma, and on top of that, you have your 4 basidiospores on average. Some basidiospores have 2, some have 2 spores, and some will have more, but the average, the main common one, is 4. Okay, so let's look at how this works. All right, so the catapult is really interesting. I suppose the history of this too is how this mechanism works. It's been studied since mid-19th century. It was first looked at by a scientist called Schmidt, and he guessed that there was something pretty unusual happening. And then it wasn't to 1909 that Buller, it was A.H. Reginald Buller, who has been described as the Einstein of mycology, because he did most amazing research. He published like 6 volumes of research experiments, and he did a lot of work on spore discharges along with a whole lot of other stuff. So in his honour, they've named this little droplet Buller's drop. So what happens, it's a catapult release driven by water tension. So what happens is the spore is not, it's not symmetrical, it's asymmetrical. So if you are studying spores of basidiomycetes and they're not they're symmetrical, it's not a blister spore. It can't be. It must be of something else that doesn't use this mechanism. Blister spore mechanism needs to have this asymmetry, and that's roughly 45 degrees that's coming off the sterigma. And what you have is right here, we call it hilar apex, right down the bottom, you have the fungus or the spore secretes mannitol, a type of sugar. It's extremely hygroscopic. What I mean by that, it makes water condense on it, okay? And in this little incurved section, while it's not symmetrical, you get other hygroscopic sugars being condensed. So what happens under high humidity, you get this droplet growing here, and you get a droplet of water growing here. And as they grow, what happens if you've ever seen two droplets on a surface come together? They all of a sudden go together very quickly through the surface tensions join and the two droplets join. When that happens, it completely shifts the center of gravity of that spore and it blasts off the sterigma, okay, which is also started to loosen by that stage. And they believe that it can, after that coalescence, the power of that have a velocity of 1 meter per second and acceleration of 1,000 Gs. It's rather saying faster than most other things in nature. Okay, so we're thinking it's like space shuttles and things here, haven't we, at that speed? But anyway, I'll calm you down now because it's spectacular launch. But after that, what happens? Okay, so you're thinking you've got the gills, right? And you've got the basidia with their spores in here. But what happens? Remember, we're talking over very, very small areas, right? So even though you've got a rapid velocity and acceleration, the wind, the air drag in here, because this is very still air. So I guess we're talking massive boundary layer. So the spore gets slowed down very quickly by the air drag. And so by the time it reaches the center between the two gills, or if we're talking about a pore, then it's in the center of the pore, they stop moving. And then it falls under gravity, right? So it's slowed by air resistance, falls under gravity. But this requires positive phototropism. So that means that geotropism, sorry, not phototropism. Positive geotropism means that those gills have to be vertical. If they're not vertical or pores aren't vertical, what's going to happen? The spores are going to impact the side. So if you've ever, and it's really well illustrated, I've seen it myself, you might have seen a Ganoderma, big fruiting body, on a log that might have fallen down into a creek or something and floods have pushed it over and it's rolled over and then it's no longer vertical and then you'll get a growth almost perpendicular to it. I'm sure this head's nodding. Yeah, so that's because to produce the spores, the pores must be vertical. So it's got positive geotropism to make them grow like that. But that's only part of the story. That gets it going. So what's happening as it falls down between those gills is the shape of the mushroom comes to play. So that mushroom, it's an umbrella first. Looks like an umbrella. Why? Because that whole droplet coalescence, if you've got rain droplets falling on top of it, that's going to bugger it up completely, won't it? So you have to protect from rain. It's an umbrella to do that. But you also need really high humidity, don't you? So that structure is actually going to keep that humidity very high, but because they only come up when it's wet anyway, because mushrooms need a lot of moisture for them to actually inflate out of the ground from the primordium up to the thing. But they've also found that the temperature on the top of the mushroom is different to the one, it's usually cooler than the environment and can be different to underneath. So that temperature differential is all about helping maintain that high humidity. And the other question I think is interesting, and I've only found one paper that sort of referred to it, is colour. Does colour come into it? And I think it's got to, because if you're talking about things where you've got to keep up optimal humidity and you're playing with temperature, a lot of your mushrooms that you see out in the open are what colour? White. And white tends to reflect heat. Where do you find the brown ones and the black ones? They tend to be often in deep in a forest. So I don't think it's not a question that's been answered because there's certainly no one's done the research. Why? Because we probably don't have enough mycologists and there's not enough funding to fund them to go and look at questions like this. So it's speculation. Anyway, so the spores, so the mushroom shape is an umbrella, maintains a high humidity, but even better than that, it's like the shape of a wing. And what happens with plain wings, you get turbulence. They change the way air currents are. And that's exactly what happens here. The shape breaks up where you have a lamellar instant wind to break it up to reduce turbulence, which is exactly what you need to carry those spores away. Okay, so we look at here. This diagram here shows that very well. And this is a photograph. You can see the spores and they're up into that turbulent layer. That turbulence goes up and connects with higher turbulence. Before you know it, those spores could be up very high in the atmosphere, depending on the conditions. So a recent paper has shown that they've done some research by modelling and etc. And they said that the maximum spore number for minimum biomass drives mushroom shape. And that's pretty much what you find all through nature really, that nature, or you know, all life has evolved to have optimal shape for its form and function. Okay. The distance between, they have found through modelling that the distance between the gills, the size of the final drop of the bullet's drop, and the spore size are finely coordinated to achieve maximum packing for that mechanism to work. They can see that size of the distance between gills and spore size, they said, is genetic, clearly, but I can't see why the droplet size wouldn't be genetic, because it would depend on the amount of mannitol being secreted by each spore, how big, and maybe there is some variation to the mannitol too. There could be. Again, it's probably just something no one's been able to investigate it. And the intergill distance they found needs to be proportional to the spore radius to the power of 3/2. So it's a finely evolved biomechanical device in its structure that should have most engineers getting very excited. Is that correct? Okay. All right. So, and I guess there's ecology in the fruiting size, because in the fruiting body, because you see a lot of variation, as you know, when you go out in forays. So a smaller fruiting body is not going to release as many spores. So they compensate by often having a lot more of them. Okay, I think was it Coprinopsis seminatus comes in large groups. A lot of Mycenas come in large groups. And then you think that, so it's about getting the spores out. That's what the fungus wants. That's its strategy. So another way then, so smaller ones, often they produce a lot more of them. The large fruit bodies, so you can look at something like this, an Amanita. You often see single bodies. You don't get a whole lot of Amanita together normally, unless it's Muscaria. And one large fruit body can discharge up to 30,000 spores per second. That's a phenomenal rate. But why do they need to do that? Because how long do these fruit bodies normally stay out in your garden? Day or two before it dries up. So they've got to release like buggery, which is what they're doing. But then you get longer lived fruiting bodies like Panis here. And they can discharge spores over time when the conditions are optimal. So they can stop, start. They've got their fruiting body, got the umbrella ready. It's a more woody structure. So that's a different strategy that they've got. And I suppose the brackets work really well on that too. Okay, and then you get another variation, which is really even more interesting, is the Coprinus, the Incap. So the Incap, you know, it sort of dissolves into this black inky mess, and you can collect the ink and you can even write with it. Why is it doing this? Well, the structure is a very, very tall cap, very tall, narrow cap. Now, for the spores to fall from the top of that, it's a big ask without impacting and without affecting these others, because they're not very narrow. It's not a lot of space for the gills. So what this fungus has done, the spores, the basidiospores on basidia mature from the bottom upwards. So they mature at different rates. So the very bottom ones at the bottom of the fresh cap, they mature first, they shoot their spores off, and then the whole tissue starts to autolyse. It starts to dissolve into that black ink. And so the next ones mature, fall, and then they autolyse all the way up until the whole process has happened. So that's why they autolyse like that. It's part of their strategy, because they've got a very, very tall structure. Okay. So hopefully every time you look at a fungus now and you look at the variation in shape, which is one of the characters you're going to be describing when you're trying to do your taxonomy, think about how this is going to affect the basidiospore release and what might be happening there, whether you've got a lot of gills or few gills, whether it's pores, spines, or whatever. Okay, right. The active release exposure. Now, this is another exciting one. So Pilobolus, which not only has a beautiful name, and it's got me talking about the lower fungi again, because mucorales are the lower ones I don't normally do, but this one's an exception. It's also called the hat thrower, and it uses a squirt gun approach. So it grows up, it develops this vesicle, a clear vesicle full of liquid, the sporangium on top. Remember, the sporangium refers to spores for lower fungi, not the higher ones. And it essentially bursts and then sends that little package of spores off on a burst of water. It's a squirt gun. But how it does this and how it fits into the ecology is an exciting part of the story. So we go on. Okay, Pilobolus is found on horse dung. And horse dung, if you've ever been a student of horse dung, it's not usually flat and like cow dung, which tends to spread out and be more liquid. Horse dung tends to be in little piles, doesn't it? Piles haphazardly on top of each other. Now, if you're a fungus trying to fruit on that, and you want to be able to shoot your spores off, half the time you're going to end up shooting into another piece of dung above you or something, aren't you? So these are the fruiting bodies it's very small, you see, and what they've developed is a phototropism so that they can avoid shooting onto another piece of dung or above them, because they ultimately need to shoot onto the fresh grass where another animal come along and eat them, because animals don't tend to eat where the dung is. So they've actually got to shoot some distance away. So how do they do that in this structure? And they tend to produce waves of spores of these fruiting bodies in the morning, and they've all shot by late afternoon, produce another batch. So the structure of this little device, it's a very simple but incredibly effective. So we have the structures. So this is the subsporangial vesicle, but it's clear, except there's an extra layer of protoplasm there and down here. That extra layer of protoplasm actually acts like a convex lens. So if this sporangium is not facing the light, what happens is the light gets bent until it focuses on the opposite side. And that stimulates the cells down here on the stipe to grow faster, and it straightens up. So when it's straight, the lines of the waves of light are coming in parallel, and they're being bent at the same angle to converge down here. So that's how it grows straight towards the light. It just needs light. And then what happens is this is a huge vacuole full of liquid. It has a lot of substance called glycogen in it. The enzymic change of the glycogen produces a range of hygroscopic sugars, so water-soluble sugars. That means that they absorb more moisture, so they keep absorbing more moisture through osmotic pressure. The turgor pressure then builds up. This starts to loosen up here as this gets bigger. This gets to a point where this is sort of sitting on top of that, and then the whole thing bursts. But the little sporangium carries with it a little ring of sticky mucilage and some sort of polysaccharide in this drop, and it flies away up to 1 metre or more. And so when it impacts onto a blade of grass, whatever it is, that little ring of mucilage helps it stick there. And it also keeps it moist, okay, to protect it from drying out. So, and then the next bit then for the, that's the discharge. So for the dispersal, waiting for some animal to come along and munch it up, goes in through its gut, and then it can go out the other end in the manure and continue its life cycle. Okay, okay, this is our cultural interlude. Don't tell me that we don't do arts in our mycology science. What's really interesting is a number of years ago, a dance group set up, and they're called Pylobolus, which is probably a beautiful name for a modern dance group, and who do very interesting interpretations of dance. But they did actually name themselves after the fungus, because the chap who set it up, his father worked in a lab, was a scientist. So he clearly found out about this fungus and how strong it is and how directed it is and the sort of way that it functions. And he thought, well, that's a really good sort of metaphor for what they want to do with these modern dance companies. So go Google it if you're bored one day. Okay. So here we have another active discharge. So this is eversion. So eversion is when something turns itself inside out. And this is a basidiomycete, so Sporobolus, and it's part of the Geasteraceae, so it's related to our Earth stars. And that's called the shotgun artillery cannibal fungus. And it's actually despised by people who might have a white house or a white car or something near a whole pile of mulch where it grows. So it's essentially a pretty little star-shaped thing, but this is the spore-bearing pit. It's like a little ball in it. And how does that work? And this is where they've landed. And apparently they're really sticky and they're really rotten to get off. You have to get special cleaning products sometimes to get them off. So how does this work? Okay, so again, we've got the spore bearing, or they call it the gleber in this case, is actually inside a cup. Now that cup is 6 layers of different types of tissue. You've got a palisade layer in the middle, which I sort of show there. So I'm not very good at this, my pointer, Wayne. I think the people online are getting better pointing than the people in the room. Palisade layer, but what happens is as these layers grow, one of the layers turns to gelatinous. So it completely gelatinizes. And again, that's effect through substances like glycogen changing to different sugars, absorbing water and changing turgor pressure. So it gets to a point where the adhesion of these 2 layers in the cup is really strong at the entrance. But as one of these layers gelatinizes, what happens is you get a change in turgor pressure to the point where all of a sudden it sort of pushes it straight out. It sort of flings it, and it flings it a long way. So the power in that is you can fling it up to 6 meters away, which is pretty not a bad feat. And so I put in this, I stole this off the internet, this picture of where someone has clearly pressure cleaned this house, where this be all mulch, and these cannibal fungi have been shooting their balls up at that white, because they also are phototropic. They're aiming for light. So anything white is that they aim for. And they've done an experiment. I think they've put it into a box, dark box, and they put a little bit of white at the top. And it's incredibly accurate the way that they can shoot straight up and hit wherever it's lighter coloured. So, okay, enough of the cannonball one. More active discharges explosion. I'm probably going to treat the ascomycetes pretty briefly because we haven't got time for more, but essentially the explosive discharge of ascospores is almost universal across the and characteristic of the ascomycetes. So ascomycetes, if you've forgotten, they're higher fungi, but unlike the Basidiomycetes, they produce a sac called an ascus. And inside the ascus are their spores, ascospores, and usually 8. That's not always the case, but it's generally 8. The ascospore, ascus, the little bag, can be inside a circle, a spherical, a sphere called a cleistothecium, or it could be inside a flask with a narrow neck called a paraththesium, or it could be inside a cup, which is an apothecium. And I think most people probably know the cup better through Pisiza, which you more commonly find on your forays. But the same thing is happening in each case, that sac is actually got a little layer of cytoplasm and a huge vacuole. And that vacuole has a whole lot of glycogen in it, and the spores, ascospores, are suspended in that. And as it matures, that glycogen converts to soluble sugars. Again, soluble sugars are drawing in water, osmotic changes, until it gets to the point where it raises the osmotic pressure and the whole thing bursts. Some of them actually have like a little lid at the top, and others, they just burst. So it's the same sort of mechanism. If you've got a cup, they can all mature almost at the same time. And the next one, I think, is, yeah, it's showing that sometimes the ascomycete discharge can actually happen just through humidity change, particularly in the cups, because the cups can all shoot at the same time. They're not restricted. And I've actually seen that on a foray years and years ago. People were like, you breathe across a little cup. I think it was like Plectania, one of the dark ones, and you're getting puffing up from them. Because so next time you find one of those, try it, get down and breathe your heavy breath on top of them and see what happens. See how fungi like you breathing heavy on. Okay, next one. These are Paraththesia. So again, they don't mature all at once because you've only got a narrow neck for the ascus to get through. So what happens here is the ascus grows up as it matures, pushes through, and then as it bursts its spores, it's gone. So the next one, ascus, can then grow up. So they will do it sequentially until all the asci have gone through and been expelled. Okay. Right, so that's enough of the active ones. Now let's get on to the insects, because we can't have a talk without having a phylales here, can we? So we have the whole phylales group, which are a gastromycete part. So they have co-opted insects, and they've done that by having a weird array of structures, from the stars to things that look like aliens, little nets like the coalus. But what they've done is evolve what is quite sophisticated, this slimy mass of spores in this mucilaginous mass that has really incredibly sophisticated odours that are highly attracted to flies. Okay, it takes quite a bit to have them evolve to think that they've got odours like rotting flesh to attract them. So what happens is they tend to come out of an egg. The water and turgor pressure in the morning pushes these things up. So about early morning, they've come up. This one's, the flies have eaten everything off. Some part of them will be covered in this dark, slimy, fetid mass. Flies come along, gorge themselves, have a party, fill themselves up. They clean all that mess, the slime off. They fly away, and the spores are in their gut. And when they defecate, they've passed the spores out somewhere. They've distributed somewhere, okay, for the whole cycle to start again. And I believe the spores become activated when they've been through the gut. Another interesting insect dispersal people have never heard of, probably, is ergot fungus. As many people know about ergot. This ergot is a nasty pathogen of cereal grain, and it's got two cycles, asexual and sexual cycle. So the asexual cycle is the first bit, when the cereal grains are flowering. It's when the fungus wants to optimize and spread as fast as it can. How this fungus works is I suppose I better go back and give you botany 101. As a plant photosynthesizes, it produces sugars in the leaves, and those sugars get translocated up to the ovary, where they get turned into starches. So small sugar units get packed into very large starch molecules. And that's the food reserve for the growing embryo of the seed. But what the fungus has done when it takes over, it stops that process of going from simple sugars, glucose to starch. It co-opts it, and it produces lots and lots of spores in the sugary mass. So it keeps it as a sugary mass. And of course, this is highly attractive. It's called honeydew. It's highly attractive to insects because insects are always looking for a carbohydrate meal for their energy. So you get flies, you get bees, you get all sorts of things coming and feeding off that honeydew, and they're shifting it around to other grasses. So it's a strategy for the asexual cycle to spread very, very rapidly during the flowering of these grasses, and anything from wheats through to, I mean, we have ergot on paspalum in Queensland. It's very common. I was going to do a master's on it. That's why I know a little bit about it. Long, long time ago, sometime in the 70s. Anyway, so that's an insect media. It's vectored spreading of the spores of that fungus. Because if you took that honeydew, put it underneath the microscope, it's just packed full of the asexual spores of claviceps, the name of the fungus. When it gets cool, there's a switch where the fungus stops producing the honeydew. It then starts to grow. It grows on those sugars, and it produces a hard structure called a sclerotia, and that's, or sclerotium singular, and that mimics the shape of whatever the seed would have been in that grain. If you're rye, the ryegrass, or rye, the grain that we eat, then it's usually got a long, dark, purplish structure that looks not dissimilar to, if you look at it quickly, to the rye grain. In the case of paspalum, it's actually a little round structure that looks very similar to paspalum seed. And if you know what you're looking for, you see the difference. And that sclerotia then falls to the ground, it overwinters, and then in the springtime, when the grasses should be flowering again, it's an ascomycete. It produces its structure. I think it's like a little club-like thing, not unlike the cordyceps. And then you get your Paraththesia, you get your spores being shot out that to the asexual spores come out and they reinfect the grasses and the cycle starts again. No, there was a funny story I had when I worked at when I worked at the Institute of the paspalum ergot is actually a major problem for livestock because ergot has so many active alkaloids. And why has it got them? Because it's stopping things eating it as it overwinters. That's why a lot of them got that strategy. And there are a few biochemists there who had been working on this ergot and they working on the chemistry of it. And they got all excited because they found some grain and they found what they thought were these new ergots. And the ergot structure in Queensland is very different to the rye one, which is a brown, purpley coloured. Ours are little round ones. And they got all excited and they thought, we found a whole new species and it looks like the ergot on ryegrass and it's in Queensland and they found it on this other grass. And they were going, I don't know whether they started writing a paper, but fortunately, I don't know how, they came to me, knowing I was a mycologist, hadn't been working there long, but I think it was because I had a really new microscope and they wanted me to take a photograph of it. And I looked at these things and I thought, they're a certain shape, but they're pointy at one end and pointy at the other. Ergot aren't like that. And they didn't realise I'd actually done a lot of work on ergot just after I graduated. And I said, I think if it's a new species, you're going to have to name it Claviceps mus, because it was actually mouse poo, because where they'd been storing it. So anyway, it tells you how people can get fooled if they don't sort of know the bigger picture. Okay, some more animal dispersal. Of course, nothing's complete without a picture of a very, very cute little bettong and a woylie who are eating truffles. So animal dispersal in some of our basidiomycetes. If you think back to how we talked about the blister spore, what's the key thing we need for mushrooms and blister spore release? It's water, isn't it? When you live on a dry continent, that's something that's short. So we know that a lot of our basidiomycetes in Australia have actually evolved to never fruit above the ground. They've gone hypergeous. We call them truffles or hypergeous fruiting bodies. What they've done instead is come up with a strategy of developing some really interesting odors. And that takes quite a bit of evolution to do that, if you think about it. So these little guys have learnt to follow that odor trail and dig down, and it's a major source of food for the marsupials in Australia. And of course, that research has probably only really been known for about 20, 25 years. And so that's animal dispersal. And again, they hop away, they distribute the spores. So we now know there's a really, really good, and a lot of these are the ectomycorrhizal fungi. So we've got a connection between the animals, the fungus, and the tree. So this tripartite relationship, which is extremely important. And the next animal dispersal, of course, the other way, is the truffle, the tuber. Now, tuber, this is not basidiomycete. This is an ascomycete, and this is the original truffle in Europe, where people knew for centuries that, well, for some reason, people valued these, the fruiting bodies. Again, they fruit under the ground, and they valued them, but they knew that pigs, particularly female pigs, were incredible to go and hunt for them. They could pick them up and dig down and get a huge smile on their piggy face when they found one. And why? It's only found many years later, more recent years, that the odour produced by this is actually mimics the odour of the male sex pheromone of the male pig. And that's why she gets pretty excited about when she's digging around for them. So again, fungi have evolved these really interesting strategies for either ejecting, discharge or dispersal. So that brings us to the end of that bit. Oh no, sorry, one more. I've forgotten about water dispersal. So water dispersal. Now, it makes a lot of sense that you need to be dispersed in water and float in water and find yourself a new leaf, because a lot of the water fungi, you want to stay afloat. You can't, if you're a single round spherical shape or ellipsoidal shape, not that great for carrying and catching onto things. So next time, if you're out in a foray, you take a little container. If you see a whole lot of froth on a freshwater stream, get a sample, take it back, whack it underneath the microscope. You're going to start to see these fantastical shapes. Those spores are that shape because that allows them to catch onto things and tumble through the water. We used to do it years ago because I was working, when I first started working in the botany department, with people on some aquatic fungi, and we would do this for fun when we go out, when we got sick of working in the lab. And marine fungi have similar fantastic shapes. So that's a simple thing. They've just evolved really fancy shapes to their spores to allow them to be carried in the water and catch on to new substrates. Okay. So we've talked about fruiting bodies. They're putting a lot of spores out. So yes, fungi release millions of tons of spores into the atmosphere every year. Are the consequences of this? Possibly. They seem to be a significant cause of allergy, but a very, very much overlooked or underestimated one. This paper was a review paper written only 2 years ago, in which they were saying that it's still, most people think hay fever, which is an incorrect name anyway, but it's like a bronchial asthma, is due to pollen grains. But most pollen's about in the atmosphere in spring. And a lot of this asthma is actually found more in autumn and winter. And we're going to get a lot more fruiting in autumn and winter, don't we? Early winter. So it's thought that there were studies done a long time ago. I remember when I was working in the botany department at UQ, someone was doing a research project on it, but I haven't heard much since then. So, but this paper was examining, it was a review of quite a lot of work that's been done, and saying that it is an area where there's a huge potential for a lot more work. It's also, they think the fungal spores can affect weather patterns. Why? Because they think they could be an important source of nucleating clouds above rainforests, nucleating water droplets. Why? If you think back to that blister spore, it said they produce mannitol and hygroscopic sugars, and that causes water to condense on them, and that's the droplet that then causes the ejection, the catapult. That water droplet, as it gets caught up in the turbulent layers, the water droplet will evaporate. But they did some work to find that if you put those spores into really, really high humidity, as you could get above a massive rainforest like the Amazon, you know, a lot of evaporation happening through the plants, that they actually found they were getting condensation on these. And so they proved it in their research. So it's, again, work that's been done. How do you prove it on large scale? What is the significance for our rainforest, for our planet? Another interesting thing of where fungi leave us probably more questions than answers, but again, should leave us with an awe and a reason to actually respect them totally. So a little bit more along the spores is that how do we, I mean, even sampling spores in the atmosphere is a really, really hard thing to do. You can put out a sticky slide, but they're going to impact and fall down and sediment at different rates. So it's not actually really accurate. You can put spore plates of agar out, but a lot of the spores in the air won't germinate on that growth media. A lot won't even grow on, if they're to do with lichens and they're producing spores, they will not grow on that media. And some of the ectomycorrhizal ones won't grow either. They only grow when they've been through an animal or whatever. So that's very inaccurate. There have been a number of different types of spore samplers designed to try to capture as best they can some of the microflora, the spores in the air. But of course, identifying spores, when you think about a lot of those ballista spores, there's not much variation in the shape of them, is there? Could be a little bit in the size. And I think there's one guide was written by a guy called Smith in the 90s, he did a lot of study on these, but there's not a lot of reference in them. And I can't see how molecular can work at that level of so tiny. If you're doing one spore, it would be really hard. So another type of spore sampler I came across, I thought this was interesting because you know that, in high spore loads, that we can have up to 10,000 spores per cubic meter or even more. Cubic meters, there's 10,000 spores in that. That's a lot of spores. So we are breathing them. But the interesting thing when you look at the Anderson sampler, it simulates the deposition of different sized particles in the human respiratory tract. Is that this air comes in, it goes through a series of filters, and each level they've actually got growth media and variation growth media to try and pick out what they've got. But we know that particles above 7 microns don't tend to go much past your nose, because we've got mucilage in our nose lining the passages and also hairs. They tend to trap them. But if they're 3 to 7 microns, which is a lot of spores, dry spores, then they will get down as far as your trachea, bronchi, and your bronchioles. So bronchial asthma, for people who are sensitive, can develop sensitivity and do, and it can easily be a cause of asthma. But working out people's individual sensitivity apparently is very, very difficult because there's so many different spores out there. Fortunately, only the ones which are less than 3 microns can make it right down to the alveoli. Aspergillus is one of those. Aspergillus is black mold. It's associated with a lot of compost. Aspergillus can get down there, and unfortunately, Aspergillus can grow at our body temperature and higher. And you just hope that your macrophages are working really well. So in that case, you've got your immune system protects you best. Or when you're working in those conditions where there may be a lot of spores, doesn't hurt to wear a simple mask. So, anyway, just leaving that. So I thought I'd just add that to have you think about what's out there in the atmosphere, because we're all excited about what a spore is and the sporocarps when we find them, but don't forget they're there. The fungus doesn't give a damn about us looking at them and marveling over it. It's just strategies to get as many spores out there as possible so that one can germinate in the right condition to continue its life cycle. So anyway, some acknowledgements. Okay, Professor Google helped me steal a whole lot of stuff off the internet. But and to do this talk, I really had to go back to my really old textbooks, because a lot of the modern textbooks just don't talk about this stuff. So I've used a lot of those, and I did try to reference them throughout. And some photos were my own. So, and the other thing is, if it was the modern ones, papers and books by Nicholas Bunny. If you really want to read someone generally he writes really well, he writes really humorously, but his technical information is absolutely fantastic. Anything by Nicholas Bunny, but he did write a chapter in one of the latest textbooks, The Fungi by Watkinson, Money, and got the other, Bodie. And so a lot of this, he did a lot of work on the blister spore, working out the 1000 Gs, etc. And yeah, it's taken a lot of late nights to do this talk, so put on about 5 kilos in the chocolate. Okay, thank you. Questions? The question is, This man's obsessed with the human sperm, obviously. And okay, he said that often in sperm, only about 10% are viable. So obviously not all spores are sperm produced are viable. Is this the same in fungi? Interesting question. Look, if I'm talking about the fungi I've worked with, which is Metarhizium bovarii, the insect pathogens, and why we do a lot of viability work, because when you're doing experimental work and infecting something, you want to know if you're putting on, 1 times 10 to the 10 spores per mil or something like that. You want to know how many of them are viable. We do viability counts. They're horrendously boring. I've done more than I ever care to do. And they're nearly all 100%, except Metarhizium, because you'll never get 100% because they have constitutive dormancy, and that's the thing that makes spores hard, because some of them will germinate at the same time, bovarii will, but Metarhizium, you'll never get 100%. But if you can somehow freeze the germination and give them time, you'll find they basically are. So in terms of large fruiting bodies, now I don't know, because if you're talking ectomycorrhizas, you can't actually do the work because you can't germinate them in the labs. You can use nuclear stains, viable, to see if they're viable, but they're not always that great. So I don't know. They're probably far more efficient than some poor human who's only 10%. And I'd say you better go and see a doctor quickly. Okay, next question. Yeah, sorry. The question was about marine and aquatic. And you said, okay, I'm not an expert on the marine. I've only looked very briefly. But I do know there's a lot of marine ascomycetes which cause soft rot in timber because they love the wet environment. And one of my fellow students, when I was back in UQ, he was doing an honours project looking at the marine fungi rotting mangrove wood out on Mud Island. He was the prince of Mud Island in those days. And so, yeah, it's a huge area, but there's very few people. Kendrick and Kohlmeyer, that's the textbook that was written years ago about aquatic and marine fungi. So it's not something I know a lot about, but I do know there's a hell of a lot of them out there, and they have some of them very important for wood rot too, and breaking down, cellulosic material associated with the seed. But it's worthwhile. Google it. Yeah, no, some of them are actually quite resistant to high salt. So what's that one? Spores of marine lichens are distributed in the air or water. Sorry, Vanessa, I really don't know. I've just said I don't really know a lot about marine fungi, but marine lichens, no, I don't. Another area I can go and read about one day when I'm curious. What are the main ways ectomycorrhizal spores germinate? You mentioned they need to go through an animal. Not all, some. I mean, again, in terms of that, I'm not an expert in spore physiology. This is just something I found really interesting. So, that's my rider on this, but there's a lot that I can't answer. It's mainly get you interested. So maybe some of you people go away and look all this up and start doing it and find a research project on that. What a cop out that one is. Okay. Any more questions I can't answer? Warwick. Yeah, remember that came into, I was talking about that. Oh, sorry. Warwick just asked, he talked about when he's looking at polypores, some have got very large pores, some have very small pores. And is there a relationship that in spore size? Yes. Remember I talked about, there was a paper came out fairly recently where they modeled it all. And they found that, yes, there's a relationship between, they're talking about the gill, but that would apply to pores. And so there's a relationship between the spore size and the space into which they're ejected, because it's a very, very finely evolved thing, because of course, like most organisms, they don't waste their energy putting too much space. There's not a lot of redundancy in terms of their structure. It's usually they're fairly parsimonious in terms of the way that they develop. So there would be very different relationships according to that paper. But they worked on the gills, but no one has looked at the pores, but they just assume it's the same principle. Yep. Just curious, somebody said here, hi, thank you so much. Curious, are snails important for spreading spores too? Yeah, because I think we had someone come from the museum, spoke here a few years ago, who's an expert in snails. And I certainly, we know that the Omphalotus nidiformis, the ghost fungus, is often got big chomps out of it taken by the giant land snails. So snails are, yes, important. Fungi are important food for them. They're fruiting bodies. So, and I think, sorry? That's right, Stanislaw or something. Yeah, actually, I remember reading, I don't know whether he's talk. I'm an applied mycologist. I do experiments, or I used to. I don't now. I'm retired. Yeah, no, I think spores don't carry a lot of food. They're just basically the DNA package for a lot of them. So I don't know if some larger spores may have glycogen reserves and trehalose and things like that. So have to go a little bit of nutrient with them. But yeah, it's a good question. I'm not really, they're going to carry the DNA they need. But Dave's right that, I don't know the size of genomes. Some are big and some are small. Okay. One last question. Question is, you're producing a lot of excess spores, and it seems as though it's a huge waste of energy or potential lot of energy. Interesting question. I don't know because spore production is not, you think about asexual spores, there's not a lot of energy. It's actually really simple. And some of them, it's simply that they just take a bit of mycelium and they just go wall it off, go bang, 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 and they've got arthrospores. So it's not, you can't liken it to what's happened. I'm sorry, that's not a pun. Can't liken it to the way we think about conventionally with animals and plants, because they are a completely different group and everything they do is different. But it's an interesting question, but I have no answer. I'm ignorant in another thing yet again. Okay, is there a way of determining their weight? Question about viable and non-viable. No, I don't think they do. I think I'm not sure if anyone has weighed a spore. I think they do have estimates, but I really don't think it would make any difference, James. But I think you can use nuclear stains that do tell you if they're alive or not. They're not 100%. I don't think they're 100% accurate for some, but yeah, that's the best way people do it. But I think weighing them with a teeny weeny tiny little balance. I mean, using a balance to 5 decimal places did my head enough. I don't know. Don't think you get anything less than that. Oh yeah, yeah. I mean, that was yeah. No, you're right. I mean, I've forgotten that's another part of my career I really loved, and it was that you weigh out spores very accurately to 5 decimal places, and then you suspend them in a given amount of liquid, and then you have to do these spore counts, and you spend a lot of time doing that with your music incredibly loud in the lab to keep yourself going. But that's how we can estimate how many spores per gram. And that is, but then that depends on the amount of moisture in the spores. Then if you take the spores and dry them down to 10% moisture, you're going to actually get a very different reading. So you need to know the percent moisture in your spores when you do the spore count. So is it sounding complicated? Yeah. There's no easy answers in nature. Um, a lot. I've probably breathed a lot too, but I'm quite normal. Okay, so is that enough? Okay, bye everybody. Thanks for listening.